Hello, George. Greetings.
Hello, how are
Um,
you?
Very good. Thank you so much for joining us
There's
today.
so many of you. I have lots of thank yous. Speaking of thank yous, these are some of the organizations that have helped bring about some of the work we're doing, um, but lots of individual thank yous to come. We have part of the context of this really is very important, which is the exponential um, journey that we're on now since the late 70s, really. Not just electronics and internet and all that, but reading and writing DNA, for example. My team has helped bring down the cost of both of those about 20 million fold with comparable improvements in quality. We'll give a little bit of background on that. So these are the four topics that I think are, our small contributions to reproduction, not just human reproduction, but also endangered species. So these will be whole genome sequencing, specifically in the context of pre-implantation genetic testing. So this is IVF. And then for IVF, we've contributed to in vitro maturation of eggs, which enables hormone reduction and so forth. And we're making little steps towards egg production as well. And then item 3 is what we can do with germline editing, cloning, xenotransplantation. Don't worry, we're not doing germline human, but we are making things that go, germline editing of things that go into humans. And you'll see that in a moment. And then finally, the most furthest out and not quite here yet, but I'll show you our progress on it, is Ectogenesis, which is long-term perfusion system for uterus. Okay, so let's start. Let's take them in order. So some of you may know something about DNA sequencing, and in particular, there's two kinds, basic kinds that are very popular now, both of which we were involved in at some level, inventing. And one of them is fluorescent next-gen sequencing, for example, Illumina and MGI. And essentially, it's just microscopy. Sometimes we call it multiplex microscopy. It started with a Nikon instrument, Nikon microscope, and it's gone all the way to back to microscopy, where we use in situ sequencing to get three-dimensional, very high resolution on all sorts of biological samples. But mainly we use it for population sequencing. And the second method is nanopore sequencing, which was a pretty wild idea at the time. I think a lot of people bet against it, which is kind of a merger, for those of you who know what a Coulter counter is, or a patch clamp. It's kind of based on those kind of things. And now, fast forward to today, there's two major flavors. One is the Oxford Nanopore, and the other is a Genia sequencing by expansion Roche system. And it all goes back to around 1987, when I started proposing it to the Department of Energy and in this patent. So what do we do with those two sequencing methods? We can impact all kinds of reproductive decision making, ranging from pre-match, before you even have any idea who you can be matchmaking, which is traditional in many cultures, and even our own. There's dating and pre-dating, which is matchmaking. And there's premarital. Doris Sharim has been practicing this very successfully now since 1985, and has almost eliminated the major genetic risk serious genetic diseases in the subpopulation, which is in the 1 to 10 million person range. The principles should be applicable to the entire planet. And then IVF is getting a little more expensive and a little more not universally applicable. Not everybody is ready for IVF, but anyway, a lot of people are, and there's this whole genome sequencing and pre-implanted genetic testing. Orchid is one of the companies that I happen to be working with. Forgot to say on the first slide here, full disclosure, in the lower right-hand corner is you can go to that website to learn about, but I'll be mentioning favorably a lot of companies, most of which I've had some connection with. Anyway, this whole genome sequencing, as opposed to partial genome or SNPs, allows hundreds of monogenic disorders that were previously undetected in embryos. Here's a list of them. So, some of the top ones are in cancer. Then there's cognitive and other brain issues, and then anatomical issues. And then, a considerable fraction of these are classified case studies as genetic. Now, if we then go to the last of the 4 stages of human development, at adults, if you've made it all the way to adult with one of these serious diseases, or even let's say a newborn being adult in the sense it's past embryo stage and fetus stage, these things now it's on the order of $4 million or more for a lifetime of care or gene therapy or surgery. And if there are false positives here, it can be medically tragic. So, for example, bilateral ovarectomy and mastectomy, you better be sure that you've got the right diagnosis. Going all the way back to the other end of the spectrum, instead of $4 million, you got on the order of sort of $100 to $300. And if it's done pre-match, there's no impact of false positives, in stark contrast. And premarital IVF is somewhere in between. So this is some of the really basic ways that genetics and these new technologies can impact reproductive decision making. So let's take one of those, the third one, and drill down a little bit on what we've done for that. This is IVF and in vitro maturation. We've made ovarian follicles or follicle-like material from stem cells, derived. Actually, some of them were derived from me as a guinea pig, but lots of others. And these are recognition that there's about almost 3 million IVF cycles done per year. People might do 5 cycles to get one birth. It varies, and the price also varies. It's not dirt cheap, but we think we can reduce the cost and pain by about 3-fold by using less hormone, but that requires maturation. Here's a couple of papers that we wrote, and in particular, Merrick Schmila and Christian Cram were instrumental in forming Ovell and Gameto based on these papers they published. One of them was published at Harvard. I'll mention others in a moment. And Merrick, I think, will be at this meeting. And so here we're using the first induced pluripotent stem, let's say, pluripotent stem cell based therapy. So this is a crowded field ever since Shinya Yamanaka did his work in 2006, got a Nobel Prize for it. There's a lot of therapies aiming for FDA, but this is the first one that I know of that's made it approved and into phase 3. And it's based on having these granulosa-like cells that produce steroids and in response to FSH, they make growth factors and mature these immature cumulus oocyte complexes. So that's one thing. And then, like I said, Merrick, I think, will be at this presenting tomorrow, and his co-author on one paper and a separate paper by Sergey Belicho I think he will be here as well. I just teased you with a couple of pictures from their work, but I'm not going to talk about it any further, but instead go on to germline cloning and transplantation. Sure. So, sort of also part of reproduction. This isn't particularly about humans right at the start here, but we formed a company called Colossal. It's at least one of its goals is with endangered species. And the red wolf has been pretty endangered to the extent that it was declared extinct in the wild in 1980. Some efforts to repopulate have been plagued by the fact that it's gone through an incredible bottleneck, really down to just a few animals, more than once. And so it got up to 120 in 2012. It's back down to 15 today. We found a new population of potential red wolf DNA or animals, and we picked this one to be cloned. This is a clone that, by the standard cloning methods, developed, for Dolly the sheep initially, but many species use this, and we depend on it routinely for some of the stuff that I'll mention in a moment. But here you have kind of a smooth pipette holding an egg in place, a dog egg, and we can put in the donor nucleus, not from a dog, but from a wolf, an endangered or nearly extinct one, and this works. And we're going to be doing a lot more of that. Now, this is back to humans. This is Tim Andrews on the operating table here. He now is the longest stable xenotransplant using technology that I'll describe in a moment. And here are the surgeons, Kawai and Riella, at Massachusetts General Hospital. They wrote a paper on this in New England Journal of Medicine earlier this year. And here's them doing the operation, where we're starting a clinical trial, which we hope to get to 50 people very soon. Tim Andrews is here having a great time of his life relative to when he was on dialysis. Most dialysis patients would love to have an alternative like the one that Tim has here. And here's Leo, the surgeon, and his wife. And it's just very gratifying to see this happen. And so what happened? Let's explain what happened here. This was a pig kidney. We're aiming at both at both addressing the shortage of organs of all types all over the world, and kidney is a great starting point. We've also done some work with liver and heart, but let's focus on kidney here. It affects 14% of U.S. adults, and most of them are unaware that they have a kidney problem that's going to lead to terminal kidney disease. There's almost 600,000 on dialysis, but only 25,000 transplants per year. So that means a lot of people are suffering on dialysis. What we did, we replaced, we knocked out 3 genes that are involved in the sugar coating on the outside of most pig cells that are very, very intensely rejected. This goes way beyond the kind of matching that you do between humans, where you're matching things like the major histocompatibility. Here, these 3 had to go because they would cause rejection in ours. And then we put in 7 genes from human. We're essentially humanizing the pig so that it doesn't have problems with blood, in particular blood clotting and complement, which is and then part of the innate immune system and inflammation systems. So we made all those human compatible. But then we also had to do 59 knockouts because the FDA, and frankly, we were as well, worried about the fact that every pig organ from every pig type produces lots of these retroviruses. And if retroviruses sounds like a scary term to you, it is. They can cause cancer and a variety of other things, and they insert all over your genome. And they do infect, they go from the pig cells to the human cells, they replicate there, and then they'll infect more and more human cells. In an immune-compromised patient, this is a real problem because they could evolve because there's not a strong enough immune system to keep them in check. And then the patient could even become tolerant to them, but then evolving into a zoonotic disease. And I think the world is pretty sick of zoonotic diseases at this point, ranging from Ebola to swine flu to bird flu to COVID-19. So anyway, we felt it was a better part of valor to knock these out. So we did these all these knockouts. It's 69 edits altogether, and that's what went into Tim Andrews, the video that I just showed you. This is thanks to Luhan Yang, who is a graduate student and postdoc in my lab, and we co-founded eGenesis in Cambridge, Massachusetts, and also to Winning Quinn and Mike Curtis at eGenesis and their team. It's a wonderful team. And like I said, we're aiming to deal with the shortage of organs, but we also want to make enhanced organs that are better at storage, resistant to DNA damage, which can lead to cancer and senescence, but also make them specifically resistant to cancer and senescence from a whole variety of sources, which has been done. All of these things have been done in animals, non-pig animals in the past, so we're just transferring that know-how into pigs. They're already some of the immunity things I've already mentioned, but in addition, they are immune to a number of human-specific pathogens, and we want to make them more and more immune. So these organs should be better than the ones you would get from a human being eventually. Our immediate goal is to make sure that they're not is to try to get them so that we don't need the immune suppressants. Now, all human-to-human transplants need immune suppressants, but we think we can, because we can engineer the germline of pigs, and we can't do that for human donors. It's a big advantage. And the last topic, and keep queuing up your questions, is ectogenesis. This is a term created by Haldane around 1924. It refers to the growth of mammalians, specifically embryos outside of the body, since most other animals can already grow outside the body: fish, amphibian, reptiles, birds, and even some mammals, the monotremes. But this is specific about eutherian mammals, and we think that this would benefit from, maybe require, but certainly benefit from, if we can do it, a long-term uterus perfusion system. And I'll show you in a moment the challenge of getting perfusion to work, blood perfusion to work on almost any organ for long periods of time. We're also studying endometrial cells, and this is part of a bigger study where we're looking for all the ways that stem cells can differentiate into all the cells of your body. And we're coming up with recipes for quickly, like 4 days, 98%, converting any stem cell into any mature cell. In fact, I kind of barely mentioned that's how we got the granulosa cells early in the talk for maturing eggs, and the way we're getting eggs at Bovell and Gameto. So Sangban Han is a student in the lab that's doing most of the work I'll mention. Kathy and her team are working on the endometrial cells and the differentiation code, and Landis-Gow has started Kangaroo Biomed to get this into human patients, although mostly what I'll be talking about has to do with pigs and elephants. So again, this has to do with endangered species, just like the wolves I mentioned. All elephant species are endangered, and we don't want to, as we give them more reproductive options, we don't want to interfere with the normal reproduction, which is quite slow. It's 22 months of gestation, very large infants, 100 kilograms, and only one calf per litter. Pigs are a bit smaller, about 100 times smaller, 9 per litter, and only 114 days for gestation. But anyway, I was setting you up earlier for how hard it is to get perfusion systems or to get survival of organs at normothermic. I mean, it's also hard to get them at any temperature, but normothermic is the way they preserve their function the best, meaning 34 to 37 degrees centigrade body temperature. The record is liver at 13 days. And these are all the references. Kidney, and then it gets shorter and shorter as we go down the list. Kidney is 3 days outside the body. And then uterus is 1 day. And it's not all of these have been given the same level of effort, but nevertheless it's daunting to know that quite a number of groups have been working on all these different tissues and none of them, and I'll explain in a moment why liver might be a special case, but they're basically, it's hard to get past 3 days or even 13 days, and we need to get to 114 days or 660 days for these two species. Now we're doing it in pig for a variety of reasons that may be obvious. The liver is great. It does have challenges. We are using whole blood right now. Actually, it's reconstituted blood where we do what we can to keep it so that it can clot but doesn't clot in a way that messes up the organ and our perfusion. That's one of the things that we do with them. The liver has all sorts of functions here shown in this little cyclic diagram. Metabolism, red blood cell decomposition, hormone production. It produces most of the most abundant proteins in the blood, and we've actually used this in gene therapy to produce rejuvenating factors, proteins in the blood by getting the liver to produce them. And it makes bile and deactivates all sorts of toxins via P450s. So we have to. So if we're keeping a liver alive, it's providing all these functions to itself. But if we're keeping some other organ alive, like the uterus, we can either have a combined uterus liver or system, or we can just replace some of the liver components with non-living filters, and that's what we do. Here's kind of a close-up of the inside of one of our devices. That in the background here, this stainless steel chamber is where the organ is, and I'll show you in the next slide that functioning organ. And then these are some of the filters that do plasma, that do dialysis for all these different small molecules, heat exchangers, oxygenators, blood clot removal, and so forth. So I'll show you what this does in practice here. This is pig uterus, and you can see that it's spontaneous myometrial contraction is occurring. It's happening better on the left than on the right, because we're just made a cannulation error here. This is some idea of what, this is the code that you would use in looking for the colors. So when we initially perfuse it, we want to see it to become almost clear here, because we're perfusing it with so-called Wisconsin fluid, which is a good way of protecting it while it's cold. And so we ship it in the cold, sometimes briefly, sometimes for many hours. If it's perfused with deoxygenated blood, it looks really dark. Okay, and so the one in the middle here that's going through this brief video is healthy. And this, by the way, this is 16x speed up video. So that's why it's the contractions are so fast. Just wrapping up here, part of what we're trying to do here is help this endangered species of elephants. We we've worked with Paul Ling's group at Baylor in Texas. They have single-handedly, more or less, developed a subunit vaccine for glycoprotein, but even more significant, that was in 2022, more significantly, they've come up with a messenger RNA vaccine, somewhat inspired by the success of messenger RNA vaccines in humans. This is against this EEHV, which is a herpes virus, which kills babies at weaning, so that they're protected to some extent. Almost all the elephants have been exposed to EEHV. About 25% of them die, so this is an extinction level problem with them. And they die, the maternal antibodies protect them, but then on weaning they have a very high probability of dying, and there's no cure for them yet, but we do have this messenger RNA vaccine that looks very promising. And that was partially supported by the International Elephant Foundation and Colossal Biosciences, which I already mentioned in the context of the red wolf. We've published 2 papers on making pluripotent stem cells and dealing with this very curious phenomenon of elephants are remarkably resistant to cancer. So some mice typically die of cancer in a year or 2, and elephants, which have a lot more cell divisions, a lot more opportunity for the cell division to go awry and start dividing without control in cancer. Nevertheless, they have extremely low cancer. This is sometimes called Peto's paradox. Anyway, we've studied the instead of one protein called TP53, it has 29 copies, extra copies of TP53, slightly mutant, and so we've been figuring that out. And this is Paul with an elephant, and this is me with an elephant. And that's it. We do have a lot of clinical trials. I've mentioned some of them along the way. Some of them are not relevant to today's talk. And like I said, I have a lot of thank yous to make. Most of the human experiments were done with, including sequencing, technology development, and so forth, were done with personalgenomes.org cells, including now the world's 2 only absolutely complete perfect genome sequences are from these volunteers. My sister is in this crowd. They're the 2 of us. And then I also thank all the alumni from my lab. This is not my lab. These are about 200 alumni that showed up one day without me knowing it. And that's it. So I'm going to stop sharing and open it up for questions.
Thank you. Okay, so my first question. So you, at least 2 of your projects involve doing a fairly large number of genetic edits. So there's the red fox, the work with Colossal for de-extinction. Or sorry, not the red fox, the dire wolf.
Well, the red fox is 0 edits, but the dire wolf was 20. Yeah.
And then also the xenotransplantation.
Right.
So a lot of people here are interested in potential applications in human germline genetic engineering. And a sort of an open question, like a scientifically controversial question, is what would happen if you did sort of a large number of edits, like 50 edits, or if you did a large amount of selecting chromosomes or large DNA segments? What would happen to the phenotype? So I'm curious if you, obviously it's speculative, but I'm curious if you have guesses, like how low can you drive disease risks, or potentially how high can you increase IQ or other highly polygenic traits?
Good, albeit controversial, question. So one of the advantages we have is we have genuine reasons to be engineering germline of animals, and that's not controversial as far as I know. Not intrinsically so, the way human is. And what it does, it de-risks it. Most of the things that we consider, where people have strong ethical opinions in one part of the world or in one period of time, it changes from place to place and time to time, and we need to be cognizant of that, and we need to constantly be, measuring our approach to consensus on these topics. I think the for example, the attitude towards interracial marriage or IVF, which were called test tube babies, which was kind of a scary word. Those have changed radically during our lifetime, my lifetime anyway. So anyway, back to germline, we de-risk it by doing lots of experiments in animals, which are turning out very well. I mean, the dire wolf step function is doing well. The pigs are doing well enough that we're scaling that up and donating organs. So the question is, what happens when I think one of the advantages of germline is that you can affect every cell in the body. It's very hard to deliver gene therapies or cell therapies to every cell in the body. But you can with germline. Another advantage is you have clonality. That is to say, it goes through a bottleneck of one cell, and so you don't have a billion different experiences, a billion different cells doing a billion different things. If one of them goes awry, then you have cancer. While with this, you can check it out, make sure that everything's on target is okay, nothing off target, and then you make an animal in our case, or maybe someday a person. Then the question becomes, so let's say it's safe to go out to 69, probably hundreds, maybe thousands someday, maybe the whole genome. But anyway, safety is probably okay in terms of the editing, but there's always the safety issues of systems biology. In other words, when we change, let's say the clotting factors in the pig, we didn't know for sure that they would really humanize the pig in that regard, or adequately do so, or it might fix one thing and break another. But we're getting quite good at doing those physiological level interactions now that we have the ability to manipulate so many components genetically. So I think the simple answer is I don't know. I think it's going to be prioritized, if it's ever used at all, it's going to be prioritized by medical, at least serious diseases first, many of which are polygenic, and so the multiplex editing that we're doing is that. And I just want to say one last thing, which is that I mentioned all the advantages of germline and how it might change, but there's two big disadvantages, which is a lot of the things we want to fix in the germline are things like Alzheimer's. That's the bane of our generations and our descendants, is taking care of an aging population, many of whom are living past 90, which means almost all of them will have some kind of cognitive decline. So anyway, the clinical trial from getting from 0 to 90 is prohibitively expensive, 90 years old. While if you had something that caused, more rapid reversal of early stage cognitive decline, that would be a real winner, because, and the second thing is 8 billion of us have already missed the germline bus. That's not an option for us. So I think that combination of the speed at which you can test reversal in adults and the large market will probably mean that there'll be less enthusiasm for germline, even for non-ethical reasons, for very pragmatic reasons.
I also wonder if you, on that point, if there, one thing I'm especially interested in is, enhancing cognitive abilities, and an issue with that in terms of somatic interventions is that a lot of the important stuff has sort of already happened.
That's true. Yes, I think that's a good example of something that would be more important to do germline. I mean, taken at the opposite end of the cognitive spectrum, where people are severely disabled, like microcephaly, their brain, it just really is completely screwed up by the time they're born. So that's something you would want to fix early, and maybe at both ends of cognition, you'd want to be able to help out as much as you can. So yes, I think that will be an argument that will kick in as soon as everybody's convinced that it generally is a safe, effective, and affordable, which is one of the ethical things that we try to achieve by some of the methods I mentioned of getting the costs down.
I have one more broad question before we go to audience questions. So you talked some about some projects that you've done involving lots of like getting stem cells to differentiate into various cell types, and I'm curious for your sort of 30,000-foot view on how difficult it will be to do IVG to the quality level that would be necessary to apply it in humans, given that oogenesis and spermatogenesis are like pretty complicated processes with very, very important epigenetic reprogramming.
No, I agree. So I didn't have slides on this, but I can tell you, we have launched a couple of companies that are doing differentiation, notably GC Therapeutics. We have maybe 300 recipes that we've tested for getting various cell types, and our standards over time have gotten to the point where we try to get the differentiation to go to 98% completion in 4 days. And if it's worse than that, then we feel that we haven't finished yet. Now that won't necessarily work for all cell types, and in particular, we expected and it happened that eggs would be harder than all the other cells, and they are, and for 3 reasons that we anticipated. One was that the egg is about 1,000 times more massive than any other cell, certainly the starting cells. Number 2 is they demethylate, which I think you were referring to with the epigenetics, massively. Largely during cell divisions, you can demethylate just by not methylating, but it goes down to about 2 or 3% of its initial methylation state. And then the third thing is there's this exotic chromosome dance, chromosome mechanics of meiosis, which is very different from all the things that the other cells that we differentiate need to do. And so far we've gotten meiosis to go sort of halfway through, which is pretty remarkable, including we can get meiosis to be induced independently of oogenesis or any kind of gametogenesis, which I thought was kind of cool, and that's thanks to Merrick. And part of the reason I didn't go through that story is I think he's going to be going through it. But Ovell and Gameto are very far ahead, I think, in this applying this to humans. My lab is mainly focusing on animals.
Okay, let's move to audience questions. I guess I can repeat them. Okay, the question is, some of your companies use multiplex editing as opposed to single target editing. Is the off-target rate different for sequential single target editing versus multiplex editing?
Right, great question. So probably the one we have the most experience is with the pig that I described. The off targets are actually quite low. This is something that, when we raised the alarm about off targets, in 2013, in our very first paper on CRISPR, we acknowledged that it was low, but it becomes significant. It's on the order of 10 to the minus 6. That becomes significant when you're treating 1 billion cells in vivo gene therapy, or even ex vivo if you're not doing any screen, not doing any cloning. But with so anyway, when we're doing germline, we isolate a clone, we sequence it, and we make sure there's nothing off target and everything's correct on target. So that gives us lots of experience to answer your question, which is how much off target do we see? And the thing that involves the most simultaneous edits is knocking out the endogenous retroviruses. So that's 59 gene edits at once, and it's very rare to see off targets in that case. In fact, it was surprisingly easy in every regard. We thought it was going to be hard because they're repetitive elements, which is certainly hard to sequence. It's the bane of existence for a lot of manipulations as well. But anyway, that's easy, and we've repeated that with a large number of different repeat families that are not retroviruses. So that was good. I think the rate is low enough that if you're going to go through a clone, meaning a laboratory expansion of a stem cell or replicative cell, you won't have to worry about it because you'll sequence it and you'll make sure that's the right thing. But if you're going in conventional non-germline somatic therapy, there's a theoretical risk that has also so far nobody has died of modern gene therapy. There was a problem around the year 2000 where people got cancer from a retrovirus that was delivering the gene. We no longer use the retroviruses very much, but it landed next to the LMO2 oncogene, and that caused cancer. So, but the modern methods don't seem to have an off-target.
So the question is, have you compared the differences between doing sequential single-target editing versus multiplex when it comes to off-targets?
I would say anecdotally, this has not been, because the off-target is low enough, it's hard to do those experiments. But I would say anecdotally that if you do two sequentially, you will get twice the number of off-targets. So it'll go from 10 to the minus 6 to 2 times 10 to the minus 6. If they're all the same, meaning like one of those repeat families like the retrovirus, it's probably more like the same. It's like if you do 59 edits with one guide RNA, it's probably similar to if you did one guide RNA for one edit. Awesome. So sequential is not better than multiplex, but they're both fairly low. Yeah, it's a good question.
Thank you. So the question is, you've done cool work with the dire wolf, and you've done 20 edits. The question is, what is the limitation? Like, why can't we do 40 edits or more yet?
Yeah. Well, obviously we can do 69, and we can probably do hundreds. I think the reason we did 20 was we're trying to be as benign as possible, especially in our initial experiments. We want, I mean, there are a lot of dog lovers out there and wolf lovers, and we don't want our first, wolf to be, sick in any way. So we were very cautious in keeping the number low. And we also, and 5 of the 20 were even more cautiously chosen to be things that were already known to work in dogs or wolves. And that's because some of the alternatives were known to cause serious diseases if they're not in exactly the right combination with all the background genetics. This is what I was kind of alluding to earlier about the physiology is complicated. So I think that's the main thing that limited us is we wanted the first ones to work well and to develop our skills so that we could confidently get more. There's also ultimately there's a limit right now with how many guide RNAs can be introduced simultaneously, or we can a way we can introduce a lot at once is putting in big inserts. So we're going up to 200 kilobase inserts, which typically have maybe 200 changes that we want to make at once, and that's very efficient. That has very low off target. So that is probably the direction we will go in the future, is putting in bigger and bigger pieces of DNA, more changes at once in one piece. So again, we wanted to test the waters and get some phenotypes that we can measure. We're not done measuring the phenotypes yet for either the woolly mouse or the dire wolf, but I think that's the answer to your question.
Do you have a follow-up?
Yeah, just follow-up.
So the question is, if you had good safety profiles, how many independent edits do you think you could do? Like not the same, you're saying the same guide RNA?
Yeah.
Not the same guide RNA.
Yeah. No, I understand the question. So, well, like I said, we've done 200 at once in a big piece of DNA. We could also make big pieces of DNA that have 200 guide RNAs, but I think both methods are getting close to what we're comfortable with right now is 200 edits. I don't think that's the final limit at all. Most of these technologies are going exponential. One extreme value, just to free your imagination, is we are working on whole genome synthesis and getting that injected into cells or eggs. Just like somatic cell nuclear transfer is transferring a nucleus, we can make a pseudonucleus from an in vitro synthesized genome. That may seem like it's a long way off, but it's not. I mean, I mentioned the 20 million-fold improvement in reading and writing DNA. Here we just have a 200-fold improvement to get from the largest eukaryotic genome that's been synthesized to a pig genome. So, and that there's no real limit to the number of edits you can make other than the physiological conflicts that you have to anticipate, with AI and experiments on cells that don't involve whole organisms. So yeah, I don't think whole genome synthesis and ergo, million-fold multiplex editing is that far off. I'm not going to mention a specific date because people always love to point out how it was 5 minutes off, but anyway.
So you talked about ectogenesis, and there's both the sort of end of gestation period where in humans we can help incubate premature babies, and then there's also growing human embryos up to the 14-day stage. And the question is, in animals, what can you say about research for the early stage, past 14 days maybe? Yeah.
Right. So yes, we're getting better at preemies. We can almost get them about half of the way back, halfway through gestation backwards. And going forwards, you can get almost half for mice, say. The problem is that those two halves don't match up because going forwards, most of the methods involve kind of roller bottle type of mechanics, such that there's no formation of a placenta and umbilical cord, which you need. For example, the lamb experiments from the Flake Lab, you need that umbilical cord to properly perfuse it. So we're trying to start with a uterus rather than a roller bottle, so that by the time it progresses, it has a fully functional system. And pig uterus is really quite inexpensive, and it can support maybe up to a dozen pig embryos, and probably much more in terms of other say mouse embryos, getting all the way to term, if we can bridge that compatibility gap. For human, I think that remains to be seen exactly how we're going to be making uterus for, let's say, elephants, other endangered species, or humans. I think we'll get lots of experience with this with the animals before we get to humans, but I could be wrong. I mean, the market is certainly there for human.
Professor Church, do you have time for a few more questions?
Sure. Yeah. These are great questions. I'm enjoying the conversation.
So one thing I'm curious is you've touched on whole genome synthesis. My understanding is there was work kind of up to like 1 megabase, but there's not too much work trying to even do that or go further.
Right.
What do you think the prospects are there?
Yeah, well, the largest genome that's been synthesized, even it is not fully, the last time I checked, is not fully assembled and functional, is the yeast, which is 12 megabases. I mean, they've done so much things, interesting experiments on the way, that it's like they've made more than one genome. But anyway, technically, we don't have we're on the cusp of having the first eukaryotic genome. E. coli now, there's, a couple of synthetic E. coli genomes, which are in the 4 million base range. So you're right, and those were hard. Yeast and E. coli have been hard. I think part of the problem is that there's all these pit stops you go through where you have to do a bunch of bespoke, even if it's automated, it's still you have to go in there and make a bunch of recombinant clones and sequence them, and then pick the best one, or maybe edit them to get them right where they want to be, and you start dealing with big DNA, which has its own problems of shearing and so forth. But I think that there are pathways. There's a fairly clear road ahead where you synthesize in a way that you make fewer mistakes. I think that the people so far have been fascinated by the fact that you can get down to 99, you can get down, with an accuracy as high as 99.7%, maybe one error in 1,000, maybe even lower than that, and that's really great if you do it sequentially. But I think we can get down to error rates closer to 10 to the minus 7th, just like the same thing happened with sequencing. As we started out, we were very proud of the fact that we could get to 10 to the minus 3 error rate for sequencing, and in fact, I had a debate with people back then publicly that they said we can't promise more than 10 to the minus 2, 10 to the minus 3 for the genome project, or else it'll come in late and expensive. And I said, actually, I think if you go for higher quality, it'll be easier. And sure enough, today we get we can get 10 to the minus 10th is affordable, and 10 to the minus 7th is super cheap. It's in the order of $100 or so. So the 10 to the minus 3 is long gone. And I think that's also going to be true. So if you get it high accuracy all the way along and you don't stop the clone, then the final product is ready to go. That's my guess. But I don't think it's as far away as everybody thinks it is.
So CRISPR-Cas9 is all the rage these days, but are there other transposase or site-specific transposase or other tools that have caught your eye recently?
Well, absolutely. Actually, they caught my eye before CRISPR caught my eye. Because I should be championing CRISPR. I've benefited from it tremendously, but I've felt since the dawn of multiplex editing, which was in the early 2000s, before a decade before CRISPR, that recombinases and integrases were the way to go, and this includes transposinases and by recombinases, some of them are literally using general homologous recombination, while the integrases typically are using site-specific. And the reason these are coming back into favor is that now our ability to engineer proteins has undergone a revolution thanks to AI and these large libraries. So you can make lots of different designs and figure out the rules with AI, and then you can build almost any protein you want. So I've launched 6 companies that use AI design plus these large libraries to make various proteins, amino-acyl tRNA synthetases, viral capsids, antibodies, and so forth. And anyway, so I think we're gonna see almost all of these work, and which one wins probably a lot of them will win. It's the same thing I said about CRISPR when there were 3 companies, and people would say, you know, which one's going to win? And I say, well, I think we need more than 3. And sure enough, we do. We have more than 3 now, and they're all doing pretty well. So yes, one of the recent entries, for example, was an insertion sequence from bacteria that it was staring all of us in the face, and it uses bridging RNA. So it very different from CRISPR, but it shares in common that this easily programmed RNA. But in addition, every other transposon and the site-specific recombinase is up for grabs now because of this revolution in protein design.
So the question is, what is a library or biobank of wild-type animal DNA that doesn't necessarily exist now, but you most wish you had for these purposes?
So the question is, what genomes would I like to see sequenced, or is there? I think there's a different question there.
Yeah, what's like what sort of wide array of wild animal genomes would you like to see collected?
Yeah. So, well, part of this weighs upon. I raised the issue of endangered species and recently extinct species, which are just slightly more endangered. I think those deserve our attention. I'm prioritizing ones that are kind of easier to to justify because they play a key role in the ecosystems that matter to humans. Because after all, humans shouldn't be so self-centered and chauvinistic about their role in the universe, but they are, and so then you need to start there. And I think we shouldn't be limited to animals. There's all kinds of interesting plants and even microorganisms, some of which fall apart as a species because of the changes that happen in the keystone species, and some of them are independent of those keystone species. So I would start with the keystone species. We probably should sequence everything because sequencing is getting cheap. And it's a fairly small investment in paying the salaries for a few ecologists to go out and collect and sequence specimens, compared to the incredible risk of not understanding these complicated ecosystems. And sequencing doesn't guarantee understanding, but it's a fairly important part of not just the cataloging, but of seeing what the metabolic interactions might be by who's missing what enzyme and that sort of thing. So it's more than just cataloging. So I hope I'm not dodging your question. I'm prioritizing endangered keystone species, but also I think we can afford to do almost everything. Now that doesn't guarantee that we're going to be able to put it back together if we let things extinct. So I'm definitely a proponent of not letting things go extinct in the first place, and not rather than saying we should, we now have the ability to bring them back. We haven't proven that, and it's probably going to be expensive for a while, if not forever.
So my understanding is that you've talked about bringing back the woolly mammoth sort of as a way of having an elephant-like species that survives in the very cold climates and likes to knock down trees, and this helps with carbon for some reason.
Yep.
And then, is there another species what would your next target be after the woolly mammoth?
Well, I think if you have mega herbivores, the conventional wisdom, actually, and unconventional, both wisdom in the ecological circles that I go in, is that they need predators to keep them healthy. That seemed to be the case in Yellowstone, although there's a little bit of controversy there with the wolves being restored. But there's lots of examples in predators at every scale, from microscopic ones all the way up to mega ones. So the predators for mammoths and giant sloths and bison were the dire wolves, because they were just bigger, just big enough. They're not gigantic, but they were big enough to be bold and do that. So if we bring back the giant sloth, we've already brought back the bison from the edge of extinction. Well, it didn't go completely extinct, almost extinct in the wild, down to hundreds of animals. Now they're getting, pushing 500,000. Anyway, if we bring back the giant sloth and the elephant and the bison, then we might need some large predators, and to be done very cautiously, so that they don't interfere with cattle and humans and other large prey. I don't know if that's my number 2 after elephants. There's not a really close number 2 after that. Actually, some ethicists that I know, like Hank Greeley, want to bring back saber-toothed tiger, which is certainly a large-scale predator. I think there are a lot of birds that were on islands that were killed off by rats that were introduced by sailors and whatnot, and might be nice to have those back. I know a lot of the rare birds have aficionados that are doing their best to keep them from going extinct, but some have gone extinct, so I'm sure that would be high on people's list. The manatee has a relative that's, I think, 6 times larger, the Steller sea cow. There are a lot of whale species, but I think the main thing we need to do is we need to clean up our act in making environments for all these species. I think, for example, for the whales, if we just stopped eating the krill and feeding the krill to our cats, that would definitely improve the quality of life for a lot of endangered whale species. So
Well,
yeah.
You probably know this, but your first name is George, and the etymology of George is, the first part is geo, which is like Earth, like geometry or geography, and the second part is urge, which is, that's in the word energy. It means work. There's also the unit of ergs, and then more distantly, it's cognate with the German word, the Germanic word work. So your name means sort of farmer, but it means like earth worker. So it's appropriate that you're working the earth. You're tending to the ecosystem.
That's one of the nicest things that anybody said. Thank you.
Do we have more? Oh, okay.
I don't want this to be taken as coming from me personally, but some people were saying there was a bit of criticism with the dire wolf thing of people saying, oh well, it's fake, and that it's just like a wolf with a few edits. Are there plans though to make like a more dire version of the dire wolf that's even larger or something like that, or is he moving on to other things?
I think I heard the question, actually, in this case. Thank you for speaking up. So I've touched upon, better in this conversation than in most previous ones, I touched upon how it is that we stuck to 20. We stopped at 20 so we wanted to have a very healthy, I would call it, dire wolf 0.1, or like versions of software. It's an early version. I don't think it deserves criticism particularly. I mean, if somebody wants to bet we'll never do it, then that's interesting. I'd be happy to make a list of everybody who thinks we'll never get beyond 20, or for that matter, get beyond the million. Place your bets. But I'm pretty confident, and I thought this was a good start. It was, I think everybody would have, I think the same critics that feel that we should have done more than 20 would, if we had gotten a failed cloning, as happened with the ibex. They always trot out the ibex and how Dolly the sheep had knee problems or something. If we had trouble with cloning, then that would have been the story. And so I think I prefer to err on the humane side until we get much better at figuring out the physiology with things like, organs on chips and some sort of multiplex developmental biology in chimeras. You'll hear more about chimeras from Sergey. I think there's all kinds of ways we can humanely test hundreds, thousands of edits separately and in combinations, and then we'll be ready to take the next step. But to fuss about whether it's the other aspect of why 20 is interesting is that in many of the fields that I'm in, there's a whole exercise which is what's the minimum number. So it's like with our colleagues in the mycoplasmas and genome synthesis in general, is what's the minimum number of genes you can get away with and still call it a mycoplasm or an E. coli or something like that. And that's a very interesting question. It's more interesting than can we make an exact photocopy of an E. coli. And the same thing for their differentiated cells that we were talking about earlier. It's not only interesting to make a perfectly functioning neuron from a completely de-differentiated stem cell, it's also interesting to ask what's the minimum number of genes it takes to make that transition from pluripotent stem cell to neuron. And we've got a couple dozen recipes for doing that with one gene. Now that doesn't mean that's a perfect copy, and so haters could say, "Oh, that's not a real neuron. That's even though it has action potentials and it makes synapses with other neurons and you can make neural nets and so forth. That's not a real neuron because you only used one epigenetic factor, right?" And so, to which I say, well, actually, I think it's in our science. That's to me a more interesting achievement than making the epigenetics absolutely the same, which we will also try to asymptotically approach that. So it's a bit of pragmatism. It's a bit of showing off what the smallest number of genes we can change and still get some interesting phenotypes. So I have no regrets about making only 20 for our first attempt. I mean, it's actually pretty fast, and I think we'll be well beyond 20 soon.
Okay, the question is, there was recently a gene therapy approved for a baby that was quite quickly approved, so less than a year between when the N of 1 mutation was identified to when the drug was approved, and the FDA process was only a week long.
Yes.
And so the question is, do you see...
Have there been any fundamental changes to the FDA concerning an approval of gene editing?
Have there been any fundamental changes to the FDA regarding approval for gene editing that might be relevant to germline?
Yes.
Well, okay, there's a cluster of interesting points there. I think that baby, that case, is a milestone case. I celebrated immediately. I wrote the author, first author on it, who's a friend of mine, and congratulated him right away. And then saw him in a meeting a couple days later, coincidentally. Anyway, it's something that we've been, the field has been moving towards, which is the N of 1 study, on the premise that it actually might be less expensive, even though it seems like it could be more expensive because it's bespoke and customized. It's like a custom suit. That one, Kieran told me, was $1.5 million, and he thinks he could get it down to less than half that because the primate trials were not particularly helpful. That was not germline. That was it was a healthy baby. So in a certain sense, babies have more in common with adults in that regard, in the way that you do the gene therapy.
It was also
I think that there will be more of those. Balanced with that is that even if the cost gets down into low hundreds of thousands, there is the possibility of doing this with genetic counseling as our ability to interpret whole genome sequencing gets better, which as I mentioned in the talk, and that might be, much less expensive. But nevertheless, I think it's an amazing breakthrough. The FDA right now is prohibited from even considering, normally they would consider everything that comes in they can give it feedback on, what it would take to get that into the IND stage and then into phase 1. But anything that involves germline in the United States is they can't even recognize they received the application. Now that's not true for a lot of other countries. It's just like the rules on human cloning vary tremendously from state to state in the United States and in other countries. So, and now that's like an add-on to one of the congressional acts, and it has to be renewed, I think, every year. So that could change, but it would require some political will. In particular, there'd have to be, greatly increased sentiment that is safe and effective and affordable, which I think what we're doing with animals now, plus some compelling argument for you could do it. You have to do it in germline. Can't do it with genetic counseling, which is pre-conception, and you can't do it with somatic gene therapy, which essentially this baby was an example of somatic gene therapy. And then one last thing about the FDA, there has been slight changes in the FDA. The FDA has no control over the germline, that's congressional, but they've definitely got faster. COVID gene therapy vaccines were approved in 11 months. This one, as you said, was approved really fast. And there's a lot of things are now in the sub 3-year range, muscle treatments and so forth. So I think we're pulling into the phase that we all, the optimists at least, thought was inevitable, which is that we're getting high enough safety levels that it becomes to their advantage. And the FDA, I'm a big fan of the FDA. Not everybody knows that they're bench scientists, so most of the FDA regulators actually have a lab like mine, sort of like mine. And so they really speak our language and they sympathize, and they are not speed bumps on our way. They are really there to protect us and expedite things. So anyway, that's my little 2 bits on FDA.
Thanks. And so you developed the first direct genomic sequencing method. You also initiated the Human Genome Project, which did the first whole human genome sequence, and you started the Personal Genome Project in 2005, which was like a very early initiative to get more human genome sequenced. So I sort of assume you had like a sense of where this was going, like the explosion of like amazing science and applications. And I'm curious, this is a sort of a broad question, but I'm just curious if you have reflections on how motivations around this kind of like the technology that isn't here but could be here kind of soon and would have amazing applications. If you have any reflection on how people's motivations change, whether it's investors or scientists or regulators, et cetera.
Yeah, so it's sometimes said that the best way to predict the future is to influence it, is to make it happen. And I sort of, and a less obvious corollary to that is that I think that doing it through the government is a last resort. That the best way to do it is technologically. So if you create something really cool, very useful, safe, and so forth, then everybody will go for it, and they won't need an act of Congress. So, for example, Androids and iPhones, people would line up to get access to them. They didn't need to be reimbursed by their phone company or their medical care provider. So I think that's what we needed to do. And for sequencing, you said that it is true that I was one of the first proponents of the Human Genome Project in 1984, full 6 years before NIH got involved. The meeting was a DOE meeting, and so that's why DOE got a head start on it. But I actually quickly became a refusenik on the Genome Project, even though I helped start it, because I felt that the goal of $3 billion was the wrong goal, that the goal really should be $1,000, and it wasn't going to take longer, just like the goal of having low quality was a bad goal. I felt that high quality and low cost was actually going to be faster and cheaper in the end, and I think that's where we are today. But there was, at that time I was too young. I was just a postdoc turning into assistant professor, and I couldn't really influence things. So I just went off on my own and kept developing technology, knowing that eventually we'd be done with the $3 billion genome, and then people would say we can't keep doing it at $3 billion, and then we'd be ready. In the 90s, when we were just starting to get some of the old-fashioned sequencing done, we already had the beginnings of next-gen sequencing. So I think that's, for those of you who like technology development, I think that's the way to change the world in a positive way, rather than lobbying Congress. Although that's you can do that too if you're into it. Yeah, I don't know if that completely answers the question, but be happy to answer variations on that.
That was good. I'm happy with that. Thank you. Any last questions? No. Okay. Professor Church, we're very grateful. This was fascinating and very interesting and inspiring. Thank you so much for talking with us.
Yeah, my pleasure. Great crowd, good questions. Thank you.
Thanks.